Hello there, and welcome to the Sleepy Science Channel. Tonight, we drift into a world that is both ancient and invisible. A realm so small, it reshapes everything we think we know about life. Microbes move through every corner of our planet. From the quiet air around us to the deepest hidden layers of the Earth. They transform and invade, and reveal abilities that seem to bend the rules of nature. Their story is a glimpse behind the curtain of existence, where tiny architects shape the world in ways both subtle and astonishing. Within a single droplet of water, entire ecosystems rise and fall. On the surface of a leaf, intricate networks pulse with activity. In places where nothing else can survive, microbial life quietly thrives. They create color in hot springs, carve patterns in stone, and hold ancient secrets in their delicate structures. If you enjoy these quiet journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, breathe softly. Let your eyes grow heavy, and allow your mind to rest as we explore the gentle world of microbes. Let's begin. Microbes were the first life to change Earth's atmosphere. Long before forests, insects, or oceans filled with fish, tiny cells were already reshaping the sky. Some ancient lineages discovered a way to use sunlight to split water and store its energy, releasing oxygen as a byproduct. At first, that oxygen reacted with exposed minerals, staining ancient rocks with vivid bands of rust that still appear in cliffs today. Gradually, the gas began to accumulate in the air and dissolve into the oceans, turning a world that had been friendly to very different kinds of metabolism into a place where oxygen-breathing life could thrive. Many earlier microbial communities likely retreated into special niches as the chemistry around them changed. Others adapted and invented new ways to cope with the reactive newcomer. Over immense stretches of time, that invisible change in the sky set the stage for complex bodies, large brains, and everything we associate with an oxygen-rich world, all sparked by microscopic photosynthesizers. Some microbes can breathe metal. For certain bacterial cells, a lump of iron or manganese is more than just a rock. It is a final resting place for the electrons that spill out when they digest their food. Instead of dumping those electrons onto oxygen the way human cells do, they pass them to metal atoms in the environment, effectively breathing rust into existence. Some attach themselves to solid mineral surfaces and push electrons directly through outer membrane structures. Others release tiny molecules that shuttle charge back and forth between cell and stone. As they do this, they strip or add electrons in ways that subtly remake the minerals around them, changing whether metals stay locked in place or dissolve into water. This quiet exchange can influence the movement of contaminants, turn soluble uranium into stable solids, and help lock carbon into sediments. In laboratory settings, similar microbes are coaxed into creating electrical currents, hinting at living batteries grounded in metal-breathing metabolisms. There are microbes that eat oil as their only food. To many marine bacteria, a slick of spilled crude is less a disaster and more a sudden banquet. Their enzymes recognize long chains of carbon and hydrogen as easy pickings, clipping them into smaller and smaller fragments until the molecules can be brought inside the cell and burned for energy. Different strains specialize in different fractions, so that heavy tars, medium-weight oils, and lighter components each have their own microbial gourmets. They bloom rapidly when hydrocarbons appear, turning invisible patches of ocean into busy microscopic festivals of feeding, and then fade quietly once the food is gone. Some live naturally in oil-rich seeps that leak from the seafloor, so they are already in place when accidents release larger volumes. Their work does not erase the harm to coasts and animals, yet it steadily transforms what would otherwise linger much longer. These invisible cleaners show how microbial metabolisms can sometimes soften even human-made messes. Some microbes thrive in boiling acid pools. In places where water fizzes with volcanic gases and the air smells sharply of sulfur, pools can be hot enough to cook food and acidic enough to strip metal. Yet in that hostile mix, certain microscopic cells grow comfortably, coated in pigments that shield fragile molecules from heat and chemical attack. Their proteins are unusually stiff and stable, designed to work at temperatures where ordinary life would unravel. Their membranes are packed with special lipids that do not melt apart, even when the water around them is nearly as hot as fresh tea. These tiny chemists harvest energy from sulfur or iron rather than from sunlight, turning deadly chemistry into a gentle internal current that powers their lives. They hint that life may have begun in places that look more like cauldrons than gardens, and their enzymes quietly support modern industries that need chemistry to keep working at extreme heat. Others live happily inside solid rock deep underground. Far below soil, roots, and buried fossils, there is a hidden biosphere inside the very minerals that make up the crust. In tiny pores and hairline fractures, microscopic cells cling to surfaces that look smooth and dead to human eyes. They are so cut off from the sun that they feed on energy trickling out of the planet itself, such as hydrogen from slow reactions between water and dark rock. Their metabolism is so patient that some may divide only once in spans of time that feel almost geological. They etch faint trails through crystal lattices as they pull out nutrients, leaving signatures that geologists can recognize long after the microbes have moved on. This community within stone keeps elements cycling in places where nothing else can reach, softening the line between living matter and the solid earth. It suggests that many other rocky worlds could harbor quiet life hidden beneath their surfaces. Certain microbes survive intense radiation that would destroy most life. Some bacterial cells can endure blasts of radiation strong enough to shatter chromosomes into many fragments, a level of damage that would be instantly lethal to typical organisms. Their trick lies not in avoiding harm, but in repairing it with astonishing efficiency. The DNA inside these cells is often present in multiple copies that are tightly packed and gently intertwined, like a careful archive prepared for disaster. When energy rips through and breaks the strands, swarms of repair proteins begin reassembling the correct sequence by comparing damaged pieces across copies. The cells also stockpile antioxidants and protective molecules that limit collateral damage to proteins and membranes. They often evolved this toughness in environments where cycles of drying and rehydration can also break DNA, so radiation resistance arrived as a powerful side effect. Their survival suggests that life can persist in places exposed to high-energy particles, such as certain nuclear sites or surfaces battered by cosmic rays. Some microbes can hibernate for millions of years and wake up again. In salt crystals collected from ancient deposits, tiny cells have been found sealed inside microscopic fluid pockets, preserved since distant ages. Others lie trapped in deep permafrost, encased in frozen ground that has not fully melted for vast spans of time. In these conditions, metabolism slows almost to a halt. Protective sugars and special proteins stabilize membranes and DNA, allowing the internal structure of the cell to resist collapse. Water may be bound into glassy states that shield delicate components from damage. When conditions finally change, perhaps through melting or dissolution, a trickle of moisture and nutrients can be enough to restart life processes. In laboratory conditions, some of these slumbering microbes have been coaxed back into activity, slowly dividing after their extraordinary pause. Their patience stretches our understanding of what it means to be alive, blurring the boundary between life and geological history, and turning sediments and ice cores into vast, cold archives of possibility. Microbes have been found living high in Earth's upper atmosphere, far above mountain peaks where air is thin and temperatures can plunge well below freezing. Microscopic passengers ride invisible currents. They cling to specks of dust, sea salt, and soot, but are lofted from the surface, then swept across continents by winds that span the globe. In this aerial realm, ultraviolet light is fierce, and liquid water appears only in fleeting droplets as clouds form and fade. Yet some cells remain viable during these journeys, protected by pigments, hardy cell walls, and dormant states that tolerate dehydration and cold. They can act as tiny seeds for ice crystals inside clouds, encouraging water vapor to freeze around them and altering where and when precipitation falls. Storms can eventually carry them back toward the surface, releasing them into new environments far from their origin. This sky-borne microbial traffic connects distant ecosystems in subtle ways and reminds us that life does not end neatly at the ground. Some microbes can survive the vacuum of space. Experiments on the outer surfaces of spacecraft and space stations have exposed bacterial spores and other resilient cells directly to the harshness beyond the atmosphere. In that environment, there is no air. Extreme temperature swings can occur, and unfiltered solar radiation strikes without mercy. Many organisms perish quickly, yet certain forms persist if they are shielded from the most damaging wavelengths or protected inside tiny cracks. Their thick coats and dried-out interiors leave little water to be torn apart, and their DNA repair systems can sometimes cope with the damage that does occur. When returned to more gentle conditions, a fraction of these travelers resumes growth, as if waking from an unusually brutal sleep. Their persistence does not prove that life routinely moves between planets, yet it keeps that possibility alive and scientifically interesting. It also reminds engineers that microscopic hitchhikers can be surprisingly hard to eliminate from spacecraft surfaces. Microbes invented photosynthesis long before plants existed. The ability to capture light and turn it into chemical energy began in tiny cells inhabiting ancient waters, long before roots and leaves appeared. Early photosynthetic microbes built complex assemblies of pigments and proteins that could absorb specific wavelengths of sunlight and funnel that energy into reactions that move electrons. Some used compounds like hydrogen sulfide or iron rather than water as their starting point, painting early seas with unusual colors. Later, a lineage refined the use of water, splitting it and eventually releasing oxygen, a trick that changed the future of the planet. The molecular machinery these pioneers evolved became so successful that it was later borrowed through endosymbiosis, when one cell took up a photosynthetic partner that gradually became the chloroplast found in brie, modern plants, and algae. Every green leaf is therefore carrying a living echo of ancient microbial innovation, quietly maintaining a partnership that began in microscopic bodies floating in primordial light. Some microbes build intricate crystal structures as homes. In certain waters rich with dissolved minerals, tiny architects draw the ingredients they need directly from their surroundings and begin to build. Layer by layer, they coax minerals to solidify into delicate cages and branching frameworks that protect their fragile bodies. These structures can resemble tiny geometric cathedrals with repeating patterns far more precise than anything that randomness alone would create. The cells position themselves within these lattices so that light, nutrients, and gentle currents reach them, while predators and turbulence are kept at a distance. In many cases, the crystals grow only when the microbes are alive and guiding the process, suggesting a subtle form of craftsmanship rather than accidental precipitation. When the inhabitants die or move on, the crystalline homes persist as microscopic fossils that carry a record of the shapes these organisms preferred. Entire sediments are filled with these mineral signatures that silently reveal how ancient waters once flowed, and which microbes shaped those environments through their patient and mineral-rich handiwork. A few microbes can generate electricity naturally. Some microscopic cells have evolved a remarkable ability to pass electrons beyond their own membranes and into the wider world. As they digest organic molecules, they release streams of electrons that must be transferred somewhere, and instead of placing them onto dissolved chemicals, they push them outward onto metals, minerals, or even neighboring cells. The proteins in their membranes act like tiny conductive bridges that ferry charge through chains of atoms with surprising efficiency. In colony form, these cells often arrange themselves in layers so that electrons flow from deep interior cells toward the surface, creating a living electrical circuit embedded in mud or sediment. When scientists place electrodes into these environments, the microbes treat them as convenient sinks for their excess electrons, producing gentle currents that reveal their hidden power. These organisms offer a glimpse of how life can interact directly with the electrical properties of the planet, without wires or machinery, turning metabolism itself into a quiet and continuous source of energy. Certain microbes can turn toxic waste into harmless minerals. When contaminants seep into soil or spread through groundwater, specialized bacteria may already be waiting with metabolisms adapted to these dangerous molecules. They capture toxic metals or complex industrial chemicals and guide reactions that steadily strip away harmful parts, transforming them into forms that no longer move freely or threaten nearby life. Some convert mercury into less reactive compounds that settle harmlessly into sediment. Others lock uranium into solid minerals that no longer dissolve into water. Still others dismantle stubborn synthetic chemicals by slicing their molecular rings or pulling off chlorine atoms until the remaining fragments are simple enough for ordinary microbes to digest. These transformations take place quietly in the dark, often in flooded soils or forgotten pockets beneath industrial sites. Over time, entire plumes of contamination shrink as microbial communities work through them. Their natural chemistry provides a powerful reminder that even in damaged environments, life can sometimes turn adversity into safe and stable matter through persistent molecular skill. Some microbes produce natural antibiotics to defend their territory. In crowded soils and packed biofilms, competition for nutrients can be intense, so some microbes produce potent chemical weapons that target nearby rivals. These molecules seep outward into the surrounding environment and disrupt the cell walls, ribosomes, or metabolic pathways of competing species, leaving the producer with a cleaner patch of space to grow. Many of the medicines used in hospitals today were originally discovered from such microbial strategies, harvested from soil-dwelling actinomycetes or other harmless neighbors that happen to wage microscopic battles. In nature, these defensive compounds rarely wipe out rivals completely. Instead, they create shifting borders where different species hold small territories and adapt to each new chemical challenge. The arms race encourages diversity, pushing microbes to evolve surprising defenses and novel chemical tricks. This underground battleground, invisible to humans, becomes a global engine for molecular innovation that has shaped ecosystems for ages and continues to influence medical research as new molecules are uncovered. There are microbes that communicate with light signals in the depths of oceans, inside moist soils, and even within certain animal hosts. Some bacterial communities use faint glows or light pulses as messages. These cells produce specialized proteins that release photons when activated, creating a soft shimmer that can be sensed by neighbors equipped with matching receptors. When enough cells glow together, their combined light can coordinate group behaviors such as movement, colony expansion, or the release of enzymes that benefit the entire community. The glow can also attract larger organisms that unknowingly disperse the microbes to new habitats, turning bioluminescence into a quiet form of recruitment. In marine environments, entire clouds of bacteria may create subtle ribbons of light that twirl through currents, each flicker representing an exchange of information. Though dim to human eyes, these signals are precise and meaningful within their own scale. The phenomenon reveals that communication in nature extends into realms far smaller and more delicate than many would imagine. Some microbes navigate using Earth's magnetic field. Scattered through lakes, sediments, and soil water are tiny swimmers with internal chains of magnetic crystals. These crystals align with the planet's field the way a compass needle does, giving each cell a preferred direction. With the help of this internal compass, the microbe can swim downward or upward until it reaches a layer of water with the right combination of oxygen, nutrients, and light. The crystals themselves form when the cell deliberately accumulates iron and precipitates it into orderly arrangements that maximize magnetic strength. This remarkable feat of biological mineral engineering allows the organism to orient itself, even in murky environments where visual cues are limited. Entire communities follow magnetic contours that guide them into narrow zones where their metabolism works best. The trait is so consistent that fossilized crystals from ancient microbes have been used to study past magnetic fields, revealing that these tiny navigators have been using planetary magnetism as a guide for immense stretches of time. Certain microbes form living mats that behave like giant organisms. In tidal flats, hot springs, and shallow seas, layers of microbial life build themselves into thick carpets that ripple with internal organization. Each layer contains different species performing distinct roles, from capturing sunlight near the surface to breaking down complex molecules in deeper, oxygen-poor zones. These strata pass materials among themselves so smoothly that the entire mat behaves like a cooperative superstructure. When light rises or falls, or when currents shift, biochemical activity sweeps through the layers in waves, creating pulses that resemble the coordinated responses of much larger organisms. Over long periods, these mats trap sediments and minerals, building dome-shaped structures that can last for geological ages. Ancient examples of these layered communities form some of the oldest fossils known, revealing that long before complex bodies evolved, microbial collectives were already shaping shorelines and influencing ocean chemistry. Their modern relatives continue this patient work, forming living landscapes that operate through shared chemistry and collective resilience. Some microbes can rewrite their DNA on the fly. Certain cells have evolved genetic systems that rearrange sections of their own chromosomes or introduce new sequences with precision whenever environmental pressures intensify. Through specialized enzymes, these microbes cut, rotate, duplicate, or swap pieces of their genome, quickly creating new combinations of traits that may better match current conditions. The process is not random chaos. Instead, it reflects sophisticated biochemical rules that ensure essential genes remain intact while variable regions shift. This dynamic genome allows rapid testing of new metabolic pathways, surface structures, or defenses. If a combination works, descendants inherit the advantage. If it fails, another rearrangement may soon follow. Such adaptability astonishes researchers because it shows that evolution can occur not only across many generations, but also within a single lifetime as genomes reconfigure themselves. This nimble information management highlights the inventive ways microscopic life responds to uncertainty, using its own genetic code as a toolkit rather than a fixed blueprint. Microbes can swap genes across species in an instant. In watery environments, on plant surfaces, and even inside animal bodies, bits of genetic material drift between unrelated microbes with ease. Tiny circular DNA elements can carry useful traits such as new metabolic enzymes or resistance to certain toxins, and these packets leap from one cell to another through close contact, viral DAR. Intermediaries, or simple uptake from the surroundings. Once inside a new host, the genes may be expressed immediately, granting the cell instant access to abilities it never evolved on its own. This rapid gene exchange creates networks of shared potential that blur species boundaries in microbial communities. Entire populations can transform their capabilities as useful genes sweep through them. Scientists studying this process have uncovered pathways that move information across surprising distances, linking river microbes to soil bacteria or hospital strains to environmental cousins. The phenomenon reveals a communal dimension to microbial evolution, where invention is often collective and where survival depends on openness to incoming genetic gifts. Some microbes build floating cities on the ocean's surface. In sunlit waters far from shore, tiny architects gather around grains of organic debris or specks of dust that drift upward until they become trapped near the surface. On these small platforms, microbes release sticky molecules that bind particles together into larger rafts. As the structure grows, different species join in, forming layered microhabitats where each group performs its own part of a shared economy. Some capture light and release oxygen. Others feed on dissolved carbon compounds. Still others recycle nutrients that would otherwise sink away. Over time, the raft supports miniature food webs of bacteria, algae, and microscopic grazers. These floating communities can be carried for long distances by currents, quietly shaping local chemistry and even influencing carbon exchange between air and sea. To an observer with powerful instruments, they appear as tiny drifting archipelagos where life cooperates in ever-shifting configurations, held together by nothing more than sunlight, surface tension, and microbial brawl. Ingenuity. Certain microbes can make rocks form faster than nature normally would. In waterways rich in minerals, some microscopic cells accelerate the process of turning dissolved ions into solid stone. Their surfaces act as ideal landing pads where calcium, magnesium, or iron gather in dense clusters. Once attached, the ions begin to crystallize as the microbes release organic molecules that guide the growing minerals into stable shapes. Layer after layer forms until entire grains, nodules, or crusts appear far more quickly than they would without a biological helper. In coastal zones, these communities create hardened layers that stabilize sediments and influence how shorelines evolve. In caves, they leave delicate mineral curtains or branching structures where dripping water and microbial chemistry intersect. Over long periods, they contribute to the slow rise of geological formations that seem purely mineral at first glance, yet their internal organization reveals biological fingerprints. These organisms demonstrate how life can quietly steer geological processes, shaping landscapes through tiny chemical nudges repeated countless times. Some microbes can turn carbon dioxide into pure biomass more efficiently than plants. In hidden mats on riverbeds, in deep sediments, and in specialized laboratory cultures, there are cells that convert carbon dioxide into organic matter with remarkable speed. Instead of relying on chlorophyll and leaves, they use compact molecular machines that shuttle carbon through high-speed chemical loops. Their enzymes grip molecules tightly and reshape them with little wasted energy, allowing these microbes to pack more carbon into their bodies than many plants can manage under similar conditions. Some draw their power from sunlight, using pigments unlike the green ones familiar on land. Others thrive in darkness by tapping into chemical gradients that are stable and plentiful underground. In nature, their work helps recycle carbon in places where vegetation is sparse. Researchers study them for hints about ancient Earth, when microbial communities dominated global carbon cycles and set the stage for later ecosystems. Their efficiency suggests that the smallest organisms may still hold some of the most capable engines for drawing carbon from the air. A few microbes produce natural plastics inside their cells. Certain bacteria store energy by crafting large molecules that behave much like soft plastics. These polymers gather inside the cell as smooth granules, providing a reserve of fuel that can be tapped during hard times. Under the microscope, the granules look like pale beads nestled within the cytoplasm, each one built through careful assembly of simple carbon compounds. When environmental conditions improve, the microbes can reshape and digest the stored polymer, converting it into energy or building blocks for growth. Scientists have explored ways to encourage these microbes to produce large quantities of the natural polymer because it breaks down more easily in the environment than many synthetic plastics. In lakes and soils, the polymer helps microbes survive sudden shifts in nutrients, acting as a biological savings account. This ability shows how evolution can generate materials with properties that echo human inventions, yet form through gentle biochemical steps performed by living cells. There are microbes that live inside glacial ice. In the cold silence of glaciers, microscopic cells persist within pockets of trapped liquid that remain unfrozen even when temperatures drop far below what most life can tolerate. These pockets form along the boundaries between ice crystals, where salts and other impurities lower the freezing point. Inside these tiny refuges, cells eke out a living on sparse nutrients carried by ancient snowfall. Their metabolism slows to a near standstill, yet they repair subtle damage from cold and maintain the machinery needed to wake fully if conditions ever warm. Some produce protective proteins that keep ice crystals from growing into destructive shapes. Others generate pigments that shield them from ultraviolet light that filters through the ice. Over centuries, the movement of glaciers carries these quiet passengers downward and forward, locking them into a slow journey through solid ice. Their survival turns glaciers into vast time capsules that preserve microbial life from distant ages. Some microbes can repair broken DNA with extraordinary precision. When radiation, toxins, or natural wear break DNA strands, most organisms struggle to fix the damage accurately. Yet certain microbes carry elaborate repair systems that find and reconnect shattered pieces with astonishing accuracy. They keep multiple intact copies of their genome so damaged regions can be compared against preserved versions. When breaks occur, proteins gather at the site and guide fragments back into the correct order, preventing mutations that would otherwise be fatal. Additional molecules shield the repair machinery from oxidative stress, ensuring that the delicate process continues smoothly. The result is a level of genomic stability that seems almost improbable given the extreme conditions these microbes endure. Their capabilities have helped scientists understand how life might persist in harsh environments on other worlds. They also provide clues for improving human medical treatments that rely on repairing DNA. These organisms reveal a hidden mastery of molecular restoration that keeps life intact in the face of relentless damage. Certain microbes can break down nuclear waste materials. In soils surrounding contaminated sites and in deep underground tanks, specialized microbes perform chemical feats that alter highly reactive metals. They interact with elements such as uranium or technetium, changing their oxidation state so they become less soluble and less likely to spread through groundwater. These transformations occur when the microbes use the metals as part of their energy metabolism, transferring electrons that shift the chemical form of the element. Over time, the altered metals settle out of water and bind tightly to sediments, reducing the risk that they will migrate into surrounding ecosystems. Some of these microbial processes were discovered unexpectedly when contaminated areas began showing signs of natural stabilization. Researchers now study these organisms to understand how to encourage similar reactions in other locations. Their ability to tame such dangerous substances demonstrates how even the most hazardous environments can host life that quietly reshapes its surroundings. Some microbes feed on hydrogen gas rising from Earth's interior. Deep underground, where sunlight never reaches and organic material is scarce, hydrogen gas seeps upward from reactions between water and certain minerals. In this dark environment, specialized microbes gather around fractures in rock and absorb the gas as a primary source of energy. They split hydrogen into protons and electrons, channeling the electrons into biochemical pathways that power growth and repair. Because hydrogen is generated continuously by geological processes, these microbes live in an energy landscape that remains stable for ages. Their presence suggests that vast subsurface ecosystems exist where life depends on hydrogen rather than sunlight or organic detritus. This possibility expands our understanding of habitable environments on Earth and other rocky planets. Many researchers believe that similar deep communities could thrive beneath the surfaces of Mars or icy moons, where water and rock interact in comparable ways. A few microbes thrive in deep-sea vents hotter than boiling water. Along seafloor cracks where mineral-rich fluids surge upward at extreme temperatures, microbial pioneers cluster near the edges of shimmering plumes. Although the fluids can exceed the temperature at which water boils at the surface, pressure at great depth keeps the water from turning to steam. Within narrow bands where temperatures drop just enough to allow life, microbes grip mineral surfaces and harness chemical energy from hydrogen sulfide, methane, and reduced metals. Their enzymes remain stable under heat that would destroy most proteins. Their membranes contain sturdy molecules that resist melting. These microbes form the foundation for entire vent ecosystems, where larger organisms depend on their ability to transform volcanic chemicals into nourishment. The existence of such heat-loving cells suggests that early life on Earth may have emerged in similarly intense environments where geology and chemistry blend in remarkable ways. Some microbes can create perfect mineral cubes. In certain soils and aquatic habitats, microscopic cells guide the formation of mineral crystals that adopt strikingly regular shapes. Iron and sulfur compounds are among the most common materials involved. The microbes create conditions that concentrate these elements on their surfaces, then release organic molecules that encourage them to crystallize in orderly patterns. The resulting structures often appear as tiny cubes with sharp edges, each one a testament to the precise biochemical environment the microbe establishes. These crystals accumulate around the cells, sometimes forming protective layers that shield them from toxins or predators. In sediments, they contribute to layers of distinct mineral grains that reflect the presence of these organisms long after they disappear. The elegance of these crystals shows how microbes can influence the geometry of the inorganic world by controlling local chemical gradients with amazing subtlety. There are microbes that build coral-like structures without being animals. In shallow, sunlit waters, layered microbial communities can bind grains of sediment together with sticky secretions that harden over time. As the layers build upward, they form nodules and domes that resemble small corals, even though no animals are involved. Photosynthetic microbes often dominate the upper layers, capturing sunlight and releasing oxygen, while deeper layers process nutrients released from dying cells near the surface. Over years and decades, these communities create solid formations that can reshape coastlines by stabilizing sediments and altering water flow. Ancient versions of these structures, known as stromatolites, record some of the earliest ecological activity on Earth. Modern examples persist in a few protected bays and lagoons where grazing animals are scarce. They reveal how collective microbial behavior can imitate the architectural complexity of corals, building enduring forms through slow accumulation and shared biochemical effort. Some microbes produce chemicals that glow in total darkness. In deep water, damp soil, And even inside a few animal bodies, there are tiny cells that invest energy in making light-producing molecules. Inside these microbes, special enzymes bring together oxygen and an organic compound such as luciferin, and a small burst of energy escapes as a photon. The reaction is carefully controlled so the light appears as a steady shimmer rather than a chaotic flash. Different species tune their chemistry to create distinct shades, from cold blue to soft green, by altering the shape of the molecules involved. For the cell, the glow is not just decoration. It can safely bleed off excess energy from metabolism, helping to limit the formation of damaging reactive oxygen. The ability has evolved multiple times in distant lineages, which suggests that light-making chemistry is surprisingly easy for life to invent. Scientists now use these microbial systems as tools, inserting the glow genes into other cells to watch biological processes unfold through tiny points of living light. Certain microbes can survive being completely dried out. On exposed rocks, desert crusts, and plant surfaces, some microbes face long stretches without a single drop of liquid water. To endure this, they enter specialized resting states in which their internal contents are packed tightly and shielded by tough outer coats. Protective proteins wrap around DNA and enzymes, forming stable complexes that are less likely to unfold or break. Cell membranes reorganize into structures that can tolerate extreme shrinkage without tearing. During the dry period, metabolism falls to an almost imperceptible level. Yet repair systems remain ready to respond the instant moisture returns. When rain or dew finally arrives, water seeps back in and the molecular scaffolding relaxes, allowing life processes to resume. These cycles can repeat many times across the lifespan of a single cell. This talent for enduring repeated dehydration shapes the microbial communities that colonize stone, bark, and soil crusts, and helps them dominate environments that swing rapidly between wet and bone dry. Some microbes form slime networks that solve mazes. In damp leaf litter and decaying logs, certain microbial collectives spread as thin, glistening sheets that flow slowly across surfaces. Each patch of the sheet senses local conditions such as nutrient levels and physical barriers, and tiny differences in growth rate emerge. Where food is richer, the sheet thickens and branches. Where obstacles block progress, growth slows or stops. Over time, the widespread network retracts from unproductive paths and reinforces the most efficient routes between multiple food sources. When researchers place these organisms into carefully shaped channels, the advancing slime traces out patterns that match the shortest connections through the maze. No single cell holds a map. Instead, the network itself becomes a living calculation, written in varying thickness and direction of growth. Once a good configuration has formed, it can be maintained for long periods, with nutrients flowing along the established pathways like traffic on a carefully planned road system. A few microbes coordinate movements like multicellular creatures. On moist soil or submerged surfaces, some bacteria glide in groups that behave more like tissues than individual cells. Each organism releases signaling molecules into the environment, and neighbors adjust their motion based on the concentration they detect. When a gradient of nutrients appears, waves of cells surge in the same direction, then slow together when they reach an ideal zone. As food runs low, the community may begin an elaborate transition. Clusters of cells pile up, forming towering structures that look almost like fruiting bodies of fungi. Within these bodies, a fraction of the population transforms into hardy spores, while others sacrifice themselves to support the architecture. The whole sequence unfolds with choreography that depends on communication, timing, and differentiation. Although every participant is a single cell, their shared behavior produces forms and life cycles that resemble those of simple multicellular organisms, hinting at possible evolutionary paths from solitary pair microbes to complex bodies. Some microbes can cloak themselves from immune systems. Inside animal hosts, certain bacteria avoid detection by constantly altering the molecular patterns on their surfaces. They shuffle the arrangement of outer membrane proteins or swap in variants encoded by silent stretches of DNA, so that antibodies that recognized yesterday's version are useless today. Others decorate themselves with chains of sugars that closely resemble the host's own tissues, causing immune cells to treat them as familiar. A few retreat into intracellular niches where immune surveillance is weaker, or adopt very low activity states in which they express minimal detectable markers. These evasive strategies allow infections to persist quietly for years, flaring up only when the host is stressed or when microbial numbers rise beyond a critical threshold. The sophistication of these cloaks reveals just how long host and microbe have been locked in an evolutionary contest, each adapting to the other in a slow, intricate dance of recognition and disguise. Certain microbes steal energy from their neighbors. In dense sediments and layered biofilms, not every microbe invests in breaking down complex food molecules. Some position themselves next to hard-working neighbors and tap directly into the electrical flow generated by their metabolism. They extend conductive pili, or outer membrane cytochromes, that make intimate contact with the surface of adjacent cells. Through these microscopic connections, electrons move from donor to thief, powering the metabolism of the recipient without any exchange of traditional nutrients. In some communities, long chains of cells link together and share electrons along extended paths, blurring the line between individual and network. Energy thieves can alter which metabolic strategies succeed by draining current from certain partners more than others. This redistribution of electrons shapes the structure of the whole community, illustrating that in the microbial world, competition can target not only food and space, but also the invisible currents that drive life's chemical reactions. Some microbes form protective bubbles around themselves. In ponds, marshes, and waterlogged soils, some microbes secrete sticky polymers that swell with water and assemble into spherical shells surrounding one or a few cells. These bubbles are soft to the touch, yet form a distinct barrier between the interior and the outside world. Inside, pH and salt levels remain relatively stable even when the surrounding environment fluctuates sharply. Harmful molecules diffuse inward only slowly, giving detoxifying enzymes more time to neutralize them before they reach sensitive structures. Predatory microbes and small grazers often find it harder to latch onto or ingest the encapsulated cells. As generations pass within a bubble, the shell can thicken and incorporate mineral particles, gaining additional rigidity. When conditions improve, the inhabitants may slip out through openings in the matrix or allow the bubble to rupture, releasing a small colony into friendlier water. These microscopic capsules show how even single cells can build personalized refuges through simple secretion and self-assembly. Microbes can alter weather by seeding ice crystals in clouds. Air currents lift countless bacteria from plant surfaces, soils, and water into the lower atmosphere, where they ride within tiny droplets that form clouds. The outer surfaces of some of these cells display proteins that encourage water molecules to align into crystalline patterns at relatively warm sub-zero temperatures. When such a cell resides in a supercooled droplet, its surface can trigger a sudden shift from liquid to ice. That event often sets off a cascade, as new ice crystals grow and collide, changing the size distribution of particles within the cloud. With enough such nucleation events, the balance tips toward precipitation. Snowflakes or raindrops begin to fall, carrying the microbial passengers back toward the surface. In this way, microscopic life becomes part of the machinery that decides when and where water returns to the ground. The influence is subtle compared with large-scale atmospheric dynamics, yet it shows that living cells participate in the physics of weather. Some microbes live inside volcanic glass. When basaltic lava pours into cold seawater or chills abruptly in the open air, it can solidify into glassy rock filled with tiny cracks and pores. Over time, seawater or groundwater seeps into these microcavities, carrying trace amounts of dissolved nutrients. Microbes colonize these spaces, attaching to the inner walls and slowly drawing energy from reactions between the glass and the infiltrating water. They may oxidize iron within the glass or reduce other elements, gradually etching delicate tunnels that radiate from their former positions. Under the microscope, these patterns form branching microgalleries that are distinct from purely chemical weathering. Samples from ancient oceanic crust show similar structures, implying that microbial glass dwellers have been active for a very long time. Their presence means that newly formed crust, which might seem barren, actually hosts a quiet biosphere that helps transfer elements from solid rock into circulating fluids. Certain microbes can break down stone monuments over time. On the surfaces of statues, temples, and gravestones, microbial films settle into tiny pits where dust and moisture collect. Some of the inhabitants release organic acids that react with minerals such as calcite and feldspar, slowly dissolving them into ions that wash away with rain. Others produce dark pigments that absorb sunlight, warming the stone slightly each day and enhancing cycles of expansion and contraction that open more microcracks. A few species bind airborne particles into crusts that trap moisture against the surface, maintaining conditions that favor continued chemical attack. Over decades and centuries, these combined activities transform sharp chisel marks into softened contours and crisp inscriptions into faint traces. Conservation scientists now study the specific communities on heritage stone, hoping to encourage less damaging species and discourage the most aggressive ones. This steady biological weathering illustrates how even seemingly inert cultural treasures are in constant conversation with the smallest forms of life. Some microbes survive extreme pressure in ocean trenches. In the deepest parts of the ocean, the weight of the water above creates pressures so intense that human-built submersibles require thick metal hulls simply to avoid collapse. Yet within the sediment and among drifting particles, microbial cells continue their quiet work. Their membranes contain unusual lipids that remain fluid under crushing force, and their proteins are shaped to fold correctly even when squeezed by surrounding molecules. Inside these cells, stabilizing compounds prevent essential enzymes from deforming, allowing metabolism to continue where almost nothing else can function. Many of these microbes rely on slow, steady chemical reactions involving methane or hydrogen to generate energy, and because nutrients fall through the water column at a gentle trickle, they grow at a pace that feels geological rather than biological. Their resilience confirms that life can inhabit a realm where pressures exceed anything found on land, and it broadens our understanding of where living systems might persist on planets with deep oceans and heavy gravitational pull. A few microbes can thrive on pure sulfur and nothing else. In volcanic regions and geothermal springs, gases bubbling from the ground leave behind bright yellow deposits of elemental sulfur. To most life forms, this material offers no nourishment, yet certain microbes attach themselves to these crumbling crystals and use sulfur as their primary energy source. They strip electrons from the mineral and funnel them into chemical pathways that produce enough fuel for growth and reproduction. As they work, they reshape the sulfur into new compounds, sometimes forming delicate crusts of sulfate minerals that hint at their presence. These microbes often live at high temperatures in acidic conditions, environments that would dissolve or denature the biological machinery of ordinary cells. Their entire physiology reflects an intimate partnership with sulfur chemistry. Because their metabolisms operate without organic carbon, sunlight, or oxygen, they provide powerful models for understanding how early life on Earth may have survived before ecosystems became complex. Certain microbes build stiff armor plates around their bodies. Some aquatic and soil-dwelling microbes invest significant energy into producing rigid plates that lock together like tiles on a tiny suit of armor. These plates form from silica, calcium carbonate, or other minerals that the cells actively accumulate from their surroundings. Once inside the cell, specialized proteins guide the minerals into precise shapes, producing discs, scales, or star-like forms with striking symmetry. The finished plates assemble into protective shells that shield the microbe from predators, turbulent water, and sudden shifts in salinity. The intricate geometry of these shells is so consistent that scientists can identify species long after the cells themselves have decayed, simply by examining the remaining mineral skeletons. In some ecosystems, the accumulation of abandoned shells contributes to sediments that eventually form geological strata. The existence of these natural armors shows how even single-celled organisms can engineer complex defensive structures using nothing more than minerals, proteins, and patient self-assembly. Some microbes can move without muscles by altering their shape. Without limbs or cilia, certain microbes glide across surfaces using cycles of expansion and contraction that ripple through their membranes. Inside these cells, networks of proteins push outward in one region while retracting in another, creating a slow yet steady flow that carries the organism forward. Chemical gradients guide these motions, allowing the cell to steer toward nutrient-rich areas or away from toxins. Some species leave thin trails of slime that reduce friction, letting their flexible bodies slip smoothly across leaves, sediments, and biofilms. Others extend temporary pseudopods that grip the environment before pulling the rest of the cell forward. Although these movements occur at tiny scales, they demonstrate impressive coordination within a single microscopic compartment. The strategies reveal that locomotion does not require dedicated engines or muscular systems. It can emerge from dynamic rearrangements of the cell's own architecture, guided by careful chemical signaling and subtle shifts in internal pressure. A few microbes hunt other microbes with harpoon-like structures. Some predatory bacteria manufacture spring-loaded molecular devices that launch sharp protein needles into neighboring cells. These harpoon structures assemble inside the predator's body as tightly coiled tubes capped with pointed tips. When triggered by contact or chemical cues, the apparatus extends with explosive speed, piercing the outer membrane of nearby microbes. Through the hollow center of the harpoon, the predator injects enzymes that dissolve the target's defenses or siphon nutrients from its cytoplasm. Once the attack is complete, the predator retracts or abandons the spent harpoon and begins assembling new ones for future encounters. Entire populations coordinate these attacks, especially in nutrient-poor environments where competition is fierce. The discovery of these molecular weapons has reshaped scientists' understanding of microbial warfare, proving that even organisms invisible to the human eye can build complex tools for capturing prey, defending their niches. Some microbes can turn liquid water into gel to trap their prey. Certain microbes secrete long carbohydrate chains that expand dramatically when they absorb water, turning a thin liquid film into a sticky gel. When smaller organisms swim or crawl into this region, the thickened water slows their motion until they become completely immobilized. The trap does not rely on toxins or speed. Instead, it uses simple physics to create a barrier that weaker cells cannot escape. Once the prey is held in place, enzymes diffuse through the gel and begin digesting exposed molecules, allowing the predator to feed passively. In some biofilms, entire hunting zones form where gels accumulate in layered sheets, each one maintained by a different group of microbes. These zones stabilize the community by controlling who enters and who becomes food. The use of gel traps demonstrates that even very small organisms can shape their microenvironment with sophisticated strategies that rival those of much larger predators. Certain microbes thrive in salt levels higher than any ocean. In hypersaline lakes and crystallizing salt ponds, the concentration of dissolved minerals can rise so high that ordinary cells would shrivel instantly. Yet specialized microbes flourish there, filling the vivid red, orange, and purple waters with life. Their membranes contain unique lipids that prevent water loss, and their proteins are adapted to function in environments where salt ions crowd every surface. Instead of resisting the salt, these organisms invite it in, keeping their internal fluids nearly as concentrated as the surrounding water. This unusual chemistry stabilizes their enzymes and prevents harmful osmotic shock. Many produce pigments that protect them from intense sunlight reflecting off bright salt flats. Because of their striking colors and metabolic quirks, they often shape the appearance and chemistry of extreme lakes. Their presence proves that life can persist in conditions far beyond what most organisms can endure, using strategies that completely rewrite our assumptions about habitability. Some microbes can live inside radioactive waste tanks. In storage sites where radioactive materials break down into chemically harsh mixtures, a few resilient microbes manage to colonize cracks, sediments, and thin films on container surfaces. These cells survive exposure to radiation that damages DNA by maintaining robust repair systems capable of stitching broken strands back together before lethal mutations accumulate. Their membranes resist corrosive solutions, and their metabolic pathways function even in the presence of reactive ions generated by decaying waste. While they do not neutralize all dangers, they can influence the chemistry of the waste by altering oxidation states of metals, changing solubility, and sometimes slowing the movement of contaminants. Their existence in such extreme facilities has surprised researchers, prompting studies into whether these microbes might help manage or stabilize difficult waste streams. Their survival suggests that life can infiltrate environments thought to be completely inhospitable, adapting to stresses that combine radiation, toxicity, and instability. Microbes can influence the behavior of animals from the inside. Inside gut systems, respiratory pathways, and even brain-adjacent tissues, certain microbes produce hormones, neurotransmitter-like compounds, or metabolic byproducts that alter how animals respond to their surroundings. Some release molecules that calm their hosts, encouraging steady digestion and predictable feeding habits. Others influence appetite, energy use, or patterns of movement. In insects, microbial residents may determine how individuals choose mates or which plants they prefer. In mammals, certain microbial communities can alter stress responses by modifying signals that travel along neural pathways connected to the gut. These effects arise from constant chemical conversations between microbe and host, with each partner adapting to the other. The relationship is not always beneficial. Some microbes manipulate behavior to enhance their own transmission, making hosts seek environments that help spread the microbe. This complex interplay reveals that behavior is not shaped solely by the nervous system. It is influenced by millions of microscopic companions working quietly within. Certain microbes trigger the formation of snowflakes. Among airborne dust particles and plant fragments drifting into the sky, some bacteria present special surface proteins that encourage water molecules to arrange themselves into ordered ice. These cells often come from leaf surfaces, where they evolved mechanisms to induce frost that releases nutrients from damaged plant tissues. Once lofted into clouds, the same proteins prompt supercooled droplets to freeze rapidly. The newly formed ice crystals grow as vapor condenses onto them, branching outward into the intricate shapes familiar as snowflakes. As crystals become heavy enough, they fall, carrying their microbial centers back toward the ground. This process influences when and where snowfall begins, especially in regions with abundant vegetation. The microscopic origin of many snowflakes highlights the surprising reach of microbial activity. Tiny cells lifted by wind can seed the formation of ice structures that transform landscapes, refill rivers, and shape winter weather patterns across entire regions. Some microbes can digest wood more efficiently than termites. Inside decaying logs and deep within compost heaps, there are bacterial and fungal partners that can slice apart the toughest parts of wood with astonishing skill. They produce elaborate suites of enzymes that recognize cellulose, hemicellulose, and lignin, the tangled polymers that make tree trunks rigid and resistant to decay. Termites rely heavily on such microbes in their own guts, yet free-living wood specialists can sometimes work through solid timber faster than any insect colony. They release enzymes into tiny channels within the wood, then absorb the resulting sugars and aromatic fragments as food. As they advance, the once solid structure becomes lighter, spongier, and rich in pockets where other organisms can move in. In forests, this activity recycles enormous amounts of locked carbon and nutrients back into the soil, feeding new generations of plants. Without these invisible carpenters, fallen trees would pile up for far longer, and entire ecosystems would be starved of essential elements that were once held tight inside sturdy trunks. A few microbes capture sunlight with novel pigments unknown in plants. Beyond the familiar greens of chlorophyll, some microscopic cells use pigments that absorb unusual slices of the light spectrum. In certain oceans, bacteria harvest energy using molecules that appear pink, purple, or golden under special illumination. These pigments can capture light that slips past ordinary algal pigments, allowing their owners to survive in dim layers where other photosynthesizers struggle. Instead of building large internal structures like chloroplasts, these microbes embed their pigments in simple membranes, creating lean and efficient light harvesting systems. Some of these pigments double as protective shields against damaging radiation, turning sunlight into both a power source and a gentle umbrella. Scientists study these systems to understand how many different ways life can tap into starlight and to explore new designs for solar technologies. The existence of such pigments hints that photosynthetic worlds around other stars might glow with colors very different from the leafy green landscapes we know. Certain microbes can survive oxygen-free environments for their entire lives. In deep sediments, flooded soils, and the interiors of animal bodies, there are cells that never encounter free oxygen and would be poisoned by it. Their metabolisms rely on alternative electron acceptors, such as nitrate, sulfate, or carbon dioxide, which they reduce in carefully orchestrated chemical steps. Instead of burning fuel with oxygen, they use these compounds to complete their energy cycles, producing gases like nitrogen, hydrogen sulfide, or methane as byproducts. Their enzymes are exquisitely sensitive to oxidation, so they maintain interiors that remain strongly reduced, protected by strict control of incoming molecules. Many of these microbes played crucial roles on the early Earth, long before oxygen accumulated in the atmosphere, and their descendants still shape modern environments. They help recycle nutrients in wetlands, rice paddies, and lake bottoms, influencing greenhouse gas emissions and water chemistry. Their thriving communities show that oxygen is not a requirement for life, only one option among many. Some microbes form microbial forests on the ocean floor. On sediments beneath coastal waters and in the deep sea, filaments and stalks of microbial growth rise from the mud like ghostly grass. Each filament may be a chain of cells operating together, transporting electrons or nutrients along its length. Others are single cells perched at the tips of mineral stalks that they themselves have precipitated, lifting their tiny bodies into more favorable flows of water. When viewed across a broad patch of sea floor, These structures resemble sparse yet organized forests, with trunks, branches, and understory layers. Currents move between them, carrying oxygen, sulfide, and organic matter from one patch to another. Animals graze among these stands, scraping off microbial films as food. Over time, the growth and decay of these forests alter the texture of sediments and help control how chemicals move between seabed and overlying water. They represent another example of how collective microscopic life can generate landscapes, only this time built from cells and minerals rather than trees and leaves. A few microbes build massive mounds that can last for ages. In shallow marine lagoons and certain alkaline lakes, layered microbial communities trap and bind sediment grains, slowly building domes and ridges that can reach impressive sizes. Each day, photosynthetic partners near the surface create sticky substances that glue incoming particles in place. At night, other members of the community process the organic matter produced during daylight, releasing gases and nutrients that diffuse through the structure. As this cycle repeats, thin layers of new material accumulate, and the mound gradually grows upward and outward. Over centuries, these constructions can become rock-hard as minerals precipitate within their pores. Some ancient examples persist as fossilized structures that record environmental conditions from deep time, preserving patterns of growth that echo the daily lives of vanished microbes. Modern mounds show that this style of construction continues, turning small cooperative communities into persistent geological features that can shape shorelines and influence local water flow for generations. Certain microbes grew filaments longer than most animals. Among the sediments of some seafloors, researchers have discovered bacteria that arrange themselves into continuous chains many centimeters in length. Each filament is composed of countless cells lined up end to end, sharing a continuous outer sheath. Within this living cable, ions and electrons can move over distances enormous compared with typical cell dimensions. This allows cells at one end of the filament to engage in chemistry that would be impossible for isolated bacteria because the necessary partners for their reactions exist only far away. For example, one region may sit in a zone rich in sulfide, while another touches oxygenated water, and the filament effectively connects these distant chemical worlds. The result is a single organism that spans multiple microhabitats, linking reactions across space the way blood vessels connect tissues in larger animals. These extended bodies challenge simple notions of what a single cell or individual organism should look like. Some microbes create their own tiny ecosystems. Within a droplet of water or a grain of soil, some species engineer conditions that attract and support a constellation of other organisms. They may secrete exopolymers that form a gel-like matrix, transforming a simple surface into a structured habitat with channels, pockets, and gradients. Within this matrix, they leak metabolic byproducts that serve as food for secondary settlers, while drawing in minerals and organic compounds from the surrounding environment. As new inhabitants arrive, they contribute their own chemistry, further diversifying the microhabitat. Over time, the engineered patch develops layers, with different specialists occupying distinct niches only micrometers apart. The founding microbes often benefit from this biodiversity because their neighbors remove waste products or supply vitamins and growth factors. These miniature worlds demonstrate that the concept of ecosystem has meaning even at scales too small for human eyes, with keystone engineers, nutrient cycles, and community succession all unfolding in Dar. Spaces smaller than a pinhead. A few microbes store energy in molecular batteries inside their cells. Rather than relying solely on transient chemical gradients across their membranes, some microbes synthesize dense energy-rich molecules that serve as internal reserves. These compounds can include polyphosphate granules, reduced sulfur globules, or other high-energy polymers that accumulate during times of abundance. Under the microscope, they appear as bright inclusions scattered throughout the cytoplasm, each representing an investment of captured power. When conditions turn harsh or external energy sources dwindle, the cells tap into these stores, breaking the molecules apart in carefully regulated steps that release usable energy without wasting it as heat. This strategy allows them to survive sudden changes, such as light turning to darkness or nutrient pulses vanishing unexpectedly. In environments with strong daily or seasonal cycles, these molecular batteries smooth out the highs and lows, letting growth continue steadily. The approach resembles rechargeable storage technology at a microscopic scale, demonstrating convergent solutions to the universal challenge of coping with fluctuating resources. Certain microbes can turn poisonous metals into harmless forms. In wetlands, river sediments, and soils near natural mineral deposits, there are bacteria that specialize in transforming toxic metal ions into states that are less mobile and less dangerous. They may attach the metals to organic molecules that cause them to precipitate as tiny crystals, or reduce their oxidation state so they bind strongly to surrounding particles. Some assemble the metals into nanoparticles that remain locked within the cell or its immediate surroundings, effectively trapping them away from larger organisms. Others pass the metals along a chain of reactions that ends with incorporation into stable mineral lattices. These processes help plants and animals by lowering the concentration of dissolved toxins such as chromium or arsenic in local waters. In some cases, the microbes gain energy from these reactions, turning detoxification into a source of metabolic power. Their quiet work influences the natural distribution of metals across landscapes, protecting ecosystems in ways that usually go unnoticed. Some microbes live inside the stomachs of herbivores and enable digestion. Cows, deer, and many other plant-eating animals rely on dense microbial communities in specialized stomach chambers to access the energy stored within tough plant fibers. The microbes produce enzymes that break cellulose and related polymers into smaller sugars, then ferment those sugars into organic acids and gases. The host animal absorbs the acids as a primary energy source, effectively outsourcing the initial steps of digestion to its microscopic partners. Within these fermenting vats, bacteria, archaea, and protozoa interact in complex food webs, with one group consuming the waste products of another. Some convert nitrogen containing compounds into forms that can be reused to build protein, helping the host thrive on diets that are low in readily available nitrogen. Others handle trace compounds that might otherwise interfere with digestion. The intimate partnership is so successful that neither partner could maintain the same lifestyle alone. Herbivorous mammals walk across grasslands as enormous mobile extensions of their hidden microbial factories. A few microbes form chains that behave like electrical wires. In certain muddy seafloors and waterlogged soils, there are bacteria that link themselves into astonishing living cables. Each chain is made of countless individual cells arranged end to end, all wrapped in a shared outer covering that keeps the structure intact. Inside these cables, electrons can travel astonishing distances compared with the tiny size of any single cell. One end of the chain may sit buried deep where sulfides or other reduced compounds accumulate. The opposite end rises toward zones touched by oxygen-rich water. By connecting these two regions, the filament carries electrons like a biological power line. This ability allows chemistry that no single cell could accomplish alone, because the necessary partners for each reaction often exist far apart. The chain therefore becomes both body and conduit, a creature stretched across multiple microhabitats. When these cables are abundant, they help regulate how sediments breathe, how nutrients cycle, and how greenhouse gases move. Their existence challenges the idea that microbes must remain small and isolated, showing that some achieve immense functional size through cooperation and continuous structure. Certain microbes can break down methane before it reaches the atmosphere. Methane is a potent greenhouse gas that rises from wetlands, lake bottoms, rice paddies, and thawing permafrost. Yet in many of these places, specialized microbes quietly intercept the gas before it escapes. Some bacteria oxidize methane using oxygen from overlying water, creating harmless carbon dioxide that dissolves back into the environment. Others work in oxygen-free zones by partnering with archaea that handle different parts of the reaction, forming intricate teams that pass electrons between them with remarkable precision. These teams often flourish in sediments where methane bubbles upward from deeper layers, forming protective rings around each pocket of rising gas. As the microbes grow, they create zones where methane concentrations drop dramatically, reducing the amount that enters the air. Their activity shapes the warming potential of wetlands and freshwater ecosystems, and without them, methane emissions would be far higher. The process represents a delicate balance of chemistry and community, maintained by organisms too small to see yet powerful enough to influence the planet's climate. Some microbes can survive freezing solid and thawing repeatedly. In tundra soils, high mountain lakes, and temporary winter ponds, certain cells endure cycles of freezing and thawing that would tear ordinary membranes apart. They achieve this resilience with suites of protective compounds that prevent sharp ice crystals from forming inside their bodies. Some release proteins that bind to small patches of ice and stop them from growing into harmful blades. Others fill their interiors with sugars or alcohol-like molecules that reduce ice formation and preserve delicate structures in a flexible matrix. When temperatures fall, metabolism slows to a whisper, yet these cells continue repairing minor damage even while encased in ice. When warmth returns, they absorb water slowly, rehydrate their machinery, and resume normal life as if nothing had happened. This ability allows them to persist in environments where liquid water exists only briefly each year. It also suggests that frozen landscapes, including icy worlds beyond Earth, could shelter life capable of awakening after long, cold pauses. A few microbes maintain internal clocks that follow day and night cycles. Within certain photosynthetic bacteria, complex protein circuits switch on and off in steady rhythms that follow sunrise and sunset. Even in total darkness, these rhythms continue for many cycles, revealing that the timing comes from an internal clock rather than immediate environmental cues. The clock helps the cells prepare for changing conditions. During daylight, energy harvesting pathways rev up, pigments repair themselves, and protective systems brace against ultraviolet radiation. As darkness arrives, the cells shift to storing energy, processing waste products, and preparing components for the next day's work. This daily pattern improves efficiency and protects vulnerable machinery from nighttime stress. Researchers have transferred parts of these clocks into other organisms, demonstrating that the rhythm continues even outside its original context. The discovery that single-celled organisms keep such precise schedules expands our understanding of biological timekeeping. It shows that the concept of a circadian rhythm is not limited to plants and animals, but extends deep into the microbial world. Certain microbes can modify minerals to build stronger structures. Scattered through soils, sediments, and aquatic environments are microbes that manipulate the minerals around them, guiding the formation of harder or more stable compounds. They draw dissolved ions toward their surfaces and release organic molecules that encourage crystallization into organized patterns. Through repeated cycles of deposition, they produce coatings that reinforce nearby grains, binding loose particles into sturdy aggregates. In some cases, they precipitate iron or manganese oxides that form protective layers around biofilms, shielding them from harsh conditions. In other settings, they strengthen natural building materials by directing mineral growth along preferred paths. Over long periods, these microscopic masons influence the formation of nodules, crusts, and pavements found in deserts and shallow seas. Their mineral craftsmanship demonstrates that geology does not unfold passively. Instead, countless microbial interactions shape the physical properties of sediments, hardening them or transforming their texture through persistent biochemical nudges repeated across generations. Some microbes carry tiny magnetic crystals inside their bodies. In lakes, estuaries, and moist soils, magnetotactic bacteria build minuscule chains of magnetic crystals that act like microscopic compass needles. These crystals form inside special compartments where iron is carefully collected and shaped into uniform particles. Once aligned in a chain, the particles point the entire cell along Earth's magnetic field lines, allowing the microbe to swim efficiently toward optimal depths. Typically, these preferred zones lie between oxygen-rich surface waters and oxygen-poor, deeper layers. By following magnetic cues, the cells bypass the need to constantly sample chemical gradients, saving energy and time. The magnetic particles remain long after the cell dies, leaving trails of fossilized needles in sediments. These relics provide clues to ancient magnetic fields and record the presence of past microbial communities. The precision of these magnetic structures reveals a level of biomineral engineering that rivals the complexity of animal skeletons, condensed into a single microscopic frame. A few microbes create floating blooms visible from space. In oceans and large lakes, certain photosynthetic microbes can multiply so rapidly that they create vast swaths of color on the water's surface. These blooms, sometimes tinted turquoise, crimson, or deep jade, trace shifting patterns as currents and winds push them across great distances. Under favorable conditions, the microbes draw nutrients from upwelling waters and capture sunlight with remarkable efficiency, turning the surface layer into a living tapestry. Some blooms support entire food webs by producing oxygen and organic matter that feed zooplankton and fish. Others can become harmful when they release toxins or consume too many nutrients, destabilizing local ecosystems. Satellites track these blooms because their reach is far larger than any boat can survey. From orbit, researchers watch the dynamics of growth, decay, and dispersal, learning how tiny cells shape the productivity and chemistry of aquatic systems. These immense gatherings show that microbial life can become a planetary-scale phenomenon, visible far above cloud tops. Certain microbes produce natural sedatives in their ecosystems. Within soils, plant roots, and aquatic communities, some microbes release molecules that calm nearby organisms. These compounds may reduce stress responses in plants, quiet cellular signaling in competing microbes, or influence the chemical balance of tiny predators that feed on them. The sedative-like substances often arise as byproducts of metabolic pathways that detoxify reactive compounds or control internal pH. When these molecules diffuse into the surrounding environment, they create zones of reduced chemical tension, allowing communities to grow with less conflict. In symbiotic relationships, plants benefit from this calming chemistry because it helps roots tolerate drought, salt stress, or toxic metals. Animals that graze on microbe-rich detritus may inadvertently ingest compounds that influence their mood or behavior. Although subtle, these effects highlight the deep chemical conversations that shape ecosystems. They reveal that microbes sometimes cultivate peace within their surroundings, softening harsh conditions with quiet molecular guidance. Some microbes influence the growth of plants by releasing special chemicals. In the thin film of soil that clings to roots, microbial partners secrete compounds that shape how plants develop. Some release hormones that encourage roots to branch, increasing the plant's ability to gather water and minerals. Others produce molecules that loosen soil particles, making it easier for roots to push through dense ground. Still others defend their plant hosts by producing antimicrobial agents that ward off pathogens. The plant, in turn, leaks sugars and amino acids that nourish the microbes, forming a tightly knit partnership. When the balance is right, both partners thrive. Fields with rich microbial communities tend to support healthier vegetation, even under challenging conditions. In harsh environments, these chemical helpers can mean the difference between survival and failure. The relationship demonstrates that plant growth is never a solo act. It depends on a complex underground chorus of microscopic chemists working steadily to support towering stems and spreading leaves. A few microbes can capture nitrogen from the air and turn it into food. Although air is filled with nitrogen, most living things cannot access it directly. Certain microbes solve this problem with an enzyme system that breaks the strong triple bond holding nitrogen molecules together. Inside specialized cells or protected nodules on plant roots, these nitrogen fixers convert atmospheric nitrogen into ammonia, a form that can be built into amino acids and nucleotides. The process demands significant energy, so the microbes often partner with plants that supply sugars in exchange for nitrogen-rich compounds. In natural ecosystems, these partnerships control how quickly nutrients cycle and how productive soils can become. Some of the most fertile regions owe their richness to long-standing symbioses between microbes and plants. Without these microscopic chemists, global food webs would collapse, because nitrogen limitations would choke off growth across continents. Their work underscores the profound influence of tiny organisms on the nutrient backbone of all life. Certain microbes live in hot springs colored by their pigments, in geothermal pools where mineral-rich water surges from deep within the earth. Mats of microbes flourish in vivid bands of color. These hues come from pigments that protect cells from intense heat and ultraviolet radiation. Some pigments absorb damaging wavelengths and convert them into harmless warmth, while others help capture light for photosynthesis in water that can be close to boiling. Each band within the spring reflects a narrow temperature range because different microbial species thrive at different heat levels. As water cools while flowing outward from the source, the community gradually shifts, creating natural rainbows that trace the contours of temperature and chemistry. These microbes also influence the minerals in the pool, causing silica, sulfur, and carbonates to precipitate in complex patterns. Over long periods, their growth helps build terraces, mounds, and rims that shape the spring's iconic appearance. These colorful communities reveal that even the hottest corners of the planet are alive with carefully tuned ecosystems powered by pigments sculpted through ages of adaptation. Some microbes can break open other microbes and consume them entirely. Among the smallest predators on Earth are bacteria that specialize in invading and dismantling other cells. They slip through outer defenses by releasing enzymes that soften membranes or by squeezing through vulnerable junctions. Once inside, they reprogram the internal environment, halting vital processes and reducing resistance. As the host cell weakens, the predator deploys additional enzymes that break complex molecules into digestible fragments. It then absorbs the released nutrients, growing rapidly within the shrinking husk. When the host cell finally ruptures, new predator cells spill out, ready to attack fresh targets. This cycle is so efficient that entire populations of prey microbes can collapse in hours. In natural environments, these predators help balance microbial communities by preventing any single species from gaining unchecked dominance. Their actions also drive evolutionary innovation, since prey must continually adapt new defenses to survive. These hunters demonstrate that predation is not limited to large organisms. It thrives in realms far smaller than the eye can see. A few microbes can sense chemical trails like living navigators. Bacteria moving through water or moist soil often rely on drifting molecules to guide them toward food or away from harm. Some possess highly sensitive receptors that detect subtle gradients of amino acids, sugars, or toxins. As a cell swims, it constantly compares current conditions to those sensed a moment before, adjusting its course through short bursts of tumbling and straight runs. Over time, these small decisions create smooth movement along invisible trails. Entire populations can follow shared gradients, forming flowing streams of cells that move collectively toward nutrient-rich zones. In complex environments such as root surfaces or biofilms, these navigators weave intricate paths that thread through narrow channels with amazing precision. Their tracking ability is so refined that slight changes in chemical release from a plant root or decaying particle can redirect the flow of thousands of cells. This guidance system reveals a subtle intelligence built from simple sensors and rapid chemical comparisons repeated again and again. Certain microbes redistribute elements across entire oceans. In the vast open sea, microscopic drifters absorb trace metals, nitrogen compounds, and carbon from surrounding water. As currents move them through different depths, these cells release waste products, die, or become food for predators, scattering their elemental cargo far from where it originated. When blooms of microbes grow near the surface, they fix enormous amounts of carbon that later sink as aggregates, carrying organic matter downward into the twilight zone. Other species draw nutrients from deep water that rises during upwelling events, transporting those nutrients into surface ecosystems when they themselves drift upward. In polar regions, microbes thriving in seasonal ice release pulses of rare compounds when the ice melts, influencing food webs hundreds of kilometers away. Over long timescales, microbial movement acts as a planetary conveyor belt that shifts key elements across latitudes, depths, and water masses. This redistribution underpins the productivity of entire marine ecosystems and regulates the chemical heartbeat of the global ocean. Some microbes form layers that record ancient environmental conditions. In shallow marine environments and coastal lagoons, layered microbial mats trap sediment grains in thin sheets that accumulate year after year. Each layer preserves a snapshot of chemical conditions such as oxygen levels, salinity, and mineral availability. Some microbes produce pigments that reflect sunlight differently depending on water clarity, while others alter the ratio of stable isotopes as they metabolize carbon or sulfur. When these mats harden into rock over geological timescales, they preserve detailed traces of their former inhabitants. By studying the thickness, color, and chemistry of ancient layers, scientists can reconstruct long-vanished environments, revealing how coastlines shifted, climates changed, and early ecosystems developed. These microbial archives reach back to some of the earliest eras of life on Earth, predating fossils of complex organisms by immense spans of time. They are living diaries written in slow, patient strokes, carrying memories of worlds that no longer exist. A few microbes preserve their communities with sticky biofilms. In rivers, pipes, plant roots, and even the human mouth, microbes assemble protective layers made of secreted polymers that form a cohesive gel. This biofilm traps nutrients, anchors cells to surfaces, and creates a stable microenvironment that buffers sudden shifts in pH, salinity, or toxin levels. Inside the biofilm, channels form that allow water and dissolved gases to circulate like miniature plumbing systems. Different species occupy distinct regions within the structure, cooperating to process incoming materials and remove waste. The outermost cells often act as a shield, absorbing harmful substances and sacrificing themselves to protect deeper layers. When conditions become unfavorable, fragments of the biofilm break free and disperse, carrying entire micro-communities to new locations. These resilient structures resist physical disturbance and chemical attack, making them one of the most durable forms of microbial organization. Their stability and adaptability reveal how cooperation transforms individual cells into thriving, persistent collectives. Certain microbes change their shape to evade predators. Some microscopic cells avoid becoming prey by altering their form whenever danger arises. When grazers approach, these microbes elongate into slender filaments, making it harder for predators to engulf them. Others inflate themselves into irregular shapes, presenting awkward angles that resist ingestion. Some grow temporary spines formed from hardened proteins, turning their once smooth bodies into thorny obstacles. These transformations can occur within hours or even minutes, triggered by chemical cues released by predators or by the vibrations caused by nearby movement. When the danger passes, the microbes revert to compact shapes that conserve nutrients and allow faster growth. This shape-shifting strategy demonstrates that even single-celled organisms possess dynamic defenses. They sense threats, modify their architecture, and wait patiently until conditions improve, showing that survival can hinge on flexibility rather than strength. Some microbes use tiny whips to swim through water. Flagella, thin threads built from spiraling protein filaments, act as rotary motors that propel many microbes through liquid environments. Deep within the cell membrane, a complex molecular engine spins the flagellum, generating thrust with each rotation. By changing direction or altering rotation speed, the microbe steers through chemical gradients, seeking nutrients or escaping harmful substances. Some species possess multiple flagella that coordinate their movements for powerful bursts of speed, while others rely on a single whip that undulates with graceful waves. In viscous environments such as mucus or dense biofilms, flagellar motion adapts, pushing like a corkscrew through thick fluid. These motors operate with mechanical efficiency far beyond human engineering, reversing instantly and running continuously without overheating. The diversity of flagellar designs demonstrates evolution's ability to shape motion at microscopic scales, allowing cells to explore vast three-dimensional spaces in search of life-sustaining conditions. A few microbes regulate their temperature with unique proteins. Although microbes lack complex organs or circulatory systems, some maintain stable internal temperatures using specialized molecules that act as heat buffers. These proteins bind to metabolic enzymes, preventing them from unfolding when temperatures rise and stabilizing their shape when temperatures drop. Other proteins influence membrane fluidity by rearranging lipid components so that the membrane neither stiffens nor melts under changing conditions. This internal regulation allows certain microbes to thrive in environments where temperatures swing dramatically between day and night or between sunlit and shaded areas. In hot springs, these proteins permit cells to remain active near the upper limits of biological tolerance, while in icy waters they prevent vital reactions from freezing into inactivity. The precision of these molecular thermostats illustrates that temperature homeostasis is not limited to animals and plants. Even the smallest cells have evolved strategies to keep their inner world steady. Certain microbes cooperate to build complex colony architecture. On submerged rocks, tree bark, and soil grains, microbial colonies sometimes form towering spires, honeycomb lattices, or dome-shaped structures. These forms emerge from collective behavior rather than centralized planning. As cells divide, they communicate with chemical signals that guide neighbors to move, pause, cluster, or disperse. Some species excrete adhesive compounds that cause local clumping, while others expand outward to create supportive frameworks. Nutrient gradients further influence architecture, since cells alter their behavior depending on how much food surrounds them. The resulting structures channel water, trap minerals, and create protected niches where new members settle. Over time, these architectures become miniature cities, complete with interior chambers, ventilation pathways, and zones reserved for different tasks. When conditions shift, colonies remodel their layouts. By working together, these microbes achieve feats of construction that mimic the coordinated efforts of much larger organisms. Some microbes generate natural surfactants that change water behavior. In ponds, soils, and even thin films on plant leaves, certain microbes release molecules that dramatically alter the way water behaves. These natural surfactants lower surface tension so effectively that droplets spread farther, soak deeper into pores, and carry nutrients in patterns very different from ordinary water flow. For the microbes, the benefit is profound. The altered water film helps dissolve hydrophobic compounds that would otherwise remain inaccessible. It can also break apart competing biofilms or disrupt predator movement by softening protective coatings. Some surfactants create tiny emulsions that trap energy-rich particles, turning drifting debris into usable food. In polluted environments, microbial surfactants help degrade stubborn oils and waxes by splintering them into manageable fragments. These molecules disperse through soil and waterways with surprising persistence, influencing water distribution at microscopic and landscape scales. The ability to re-engineer water's physical properties lets these microbes extend their reach, transform local chemistry, and reshape entire microbial communities through quiet molecular craftsmanship. A few microbes thrive inside living insects. Within the tissues of beetles, aphids, termites, and countless other insects, specialized microbes take up residence as long-term partners. Some live in dedicated cells or organs created by the host specifically to house them. Others occupy gut chambers where they receive a steady diet of plant sap, wood fragments, or nectar. In exchange, they synthesize essential nutrients such as amino acids or vitamins that the insect cannot obtain from its simple diet. These internal collaborators may also detoxify plant defenses, allowing insects to feed on otherwise dangerous leaves. Some influence development, controlling whether an insect grows wings, how large it becomes, or when it reaches maturity. In extreme cases, the microbe's presence determines the insect's reproductive success. Over evolutionary stretches, hosts and microbes coadapt so tightly that neither can survive without the other. These intimate alliances reveal that insect physiology and behavior depend not only on genes coded in the insect cells, but also on a quiet community living within. Certain microbes carry out all their life functions inside a single droplet of water. Within tiny droplets suspended in air, clinging to leaves, or nestled between soil grains, microbial worlds thrive in spaces far smaller than a grain of sand. These droplets hold only trace amounts of carbon, nitrogen, and minerals, yet some bacteria and protists complete their entire life cycles within them. Cells divide, compete, communicate, and die within a sphere so small that a gentle breeze could carry it halfway across a field. To survive, these microbes develop metabolic strategies optimized for extreme scarcity. Some enter semi-dormant states between pulses of nutrients, while others scavenge every molecule drifting through their microscopic universe. When droplets collide or evaporate, communities mix or collapse, creating rapid shifts in population and chemistry. The existence of these self-contained habitats shows that life does not require lakes, puddles, or raindrops. A thin sheen of moisture is enough for ecosystems complete with predators, prey, and intricate chemical exchanges. Some microbes have genomes smaller than many viruses. Among the tiniest free living cells are bacteria, whose genetic instructions are pared down to the absolute essentials. Their genomes contain only the minimum number of genes required for metabolism, replication, and interaction with the environment. This reduction reflects extreme specialization. These microbes often inhabit nutrient-rich niches, such as animal mouths or sheltered aquatic microenvironments, where they depend on their surroundings for many compounds they no longer produce. Despite their tiny genomes, they maintain complex cellular machinery and can respond to environmental cues with surprising precision. Their existence highlights a fundamental difference between viruses and living cells. Viruses may have small genomes, but they rely entirely on host machinery to replicate. These minimalist microbes, however, remain autonomous even while operating on a genetic budget of remarkable thrift. Their tiny instruction sets challenge assumptions about how much DNA life truly needs. A few microbes grow faster than almost any other life form. Under ideal conditions, certain microbes divide at breathtaking speed, turning one cell into billions in remarkably short periods. Their rapid growth depends on streamlined metabolisms, highly efficient ribosomes, and internal organization that minimizes wasted steps. Some species carry multiple copies of their genome, so that DNA replication can begin for future generations, even before the current cell has finished dividing. Others optimize every stage of nutrient uptake, converting food directly into new cellular material with minimal energy lost as heat. In nutrient-rich environments such as compost piles, food waste, or laboratory cultures, these speed specialists dominate, overwhelming slower species and reshaping community structure. Their growth rates approach theoretical physical limits for how quickly biological molecules can assemble. This runaway productivity has practical uses in biotechnology, where such microbes become tiny factories for enzymes, vitamins, and other valuable compounds. Their astonishing pace reveals the upper extremes of life's capacity for rapid expansion. Certain microbes can capture carbon in long-lasting mineral forms. Some microbial cells influence carbon storage by guiding the formation of solid minerals that trap carbon for decades or centuries. They encourage dissolved carbon dioxide to react with calcium, magnesium, or iron, forming stable carbonates that settle into sediments. These reactions may occur on cell surfaces, within extracellular matrices, or through enzymes that regulate local pH. Over time, mineral crusts grow thick enough to protect entire microbial mats from debris. Erosion. In certain environments, these processes contribute to limestone deposits and carbonate-rich soils. By encouraging carbon to transition from gas to solid mineral, these microbes quietly help regulate atmospheric chemistry. Their work operates on scales too small for direct observation, yet accumulates into geological features that influence climate cycles. Understanding these organisms can also inspire strategies for carbon sequestration that mimic nature's patient and persistent methods. Some microbes form hollow spheres that act like protective shells. In freshwater lakes, marine environments, and even damp soils, certain microbes secrete layers of material that curve into perfectly rounded spheres. Inside these capsules, clusters of cells remain protected from predators, toxins, and sudden shifts in salinity or acidity. The spherical shape offers maximum volume for minimal surface area, conserving resources while providing sturdy defense. Some species rotate slowly within their shells, stirring nutrients and waste products through narrow channels. Others embed themselves in thick walls that function like armor, resisting pressure and abrasion. When conditions improve, openings appear that release the internal population, allowing the next generation to disperse. These spheres may persist long after their inhabitants have moved on, becoming tiny fossils that record the architecture of vanished microbial communities. Their symmetry and durability reflect a design shaped entirely by biochemistry, requiring no instructions beyond the simple rules that govern molecular assembly. A few microbes can neutralize harmful ammonia in water. In aquariums, wetlands, rice paddies, and wastewater systems, certain microbes perform a vital service by converting toxic ammonia into safer compounds. Some oxidize ammonia into nitrite, while others complete the transformation into nitrate, which plants can readily absorb. These reactions occur at rates that influence entire ecosystems, keeping ammonia from accumulating to dangerous levels. Microbes performing these transformations often form thin films on rocks, sediments, or plant roots, where flowing water supplies them with a stable stream of reactants. Their enzymes are highly specialized and sensitive to the balance of oxygen and pH, so these bacteria cluster in zones where conditions remain ideal. Without their steady work, aquatic life would suffer from ammonia spikes, particularly in environments rich in decaying organic matter. Their presence keeps water chemistry stable and supports the productivity of both natural and human-managed ecosystems. Certain microbes thrive in caves filled with toxic gases. In remote cave systems where air hangs heavy with hydrogen sulfide, carbon dioxide, or methane, specialized microbes flourish on mineral surfaces. These caves lack sunlight and often hold atmospheres that would be fatal to animals. Yet microbial life finds footholds on damp walls and in shallow pools. Some species oxidize sulfur compounds, producing sulfuric acid that slowly carves new chambers and strange formations. Others reduce carbon compounds in low oxygen pockets, creating metabolic pathways tailored for extreme chemistry. Their pigments protect them from corrosive vapors, while their enzymes remain stable in acidic or alkaline niches. Entire ecosystems arise where no plant, fungus, or insect can survive. Over time, microbial activity sculpts cave interiors, leaving fragile textures and delicate crusts made from minerals precipitated by their metabolism. These communities expand our understanding of habitability, demonstrating that life can flourish in places lit only by chemical reactions. Some microbes create threads that link entire colonies together. In aquatic biofilms and moist soils, certain microbes extend fine filamentous structures that stretch between distant clusters of cells. These threads function like communication lines, allowing nutrients, signaling molecules, and even electrons to travel across wide areas. Colonies connected by these threads synchronize growth, coordinate defensive responses, and share resources during shortages. The filaments themselves consist of proteins arranged into conductive or semiconductive strands with internal channels that permit molecular traffic. Under the microscope, these connections resemble tiny highways bridging separate microbial settlements. When threads break, the affected colonies often weaken, showing how crucial these networks are to collective survival. Over time, the connections create sprawling communities that behave less like isolated patches and more like integrated superorganisms. These elegant networks remind us that cooperation does not require size, only persistent connection. A few microbes have enzymes capable of breaking down tough plastics. In landfills, compost piles, and even along ocean shorelines, certain microbes encounter synthetic plastics that resist decay for long stretches of time. Yet a few remarkable species possess enzymes that grip long polymer chains and slice them into smaller fragments. These enzymes recognize patterns in the plastic's structure, bending and stressing the bonds until they snap. Once broken into bite-sized pieces, the fragments enter metabolic pathways where they are converted into energy and simple carbon compounds. Some microbes work on polyester-based plastics, while others attack polyurethane or similar materials that normally linger for generations. In controlled environments, these microbes can gradually soften rigid plastic films, turning them into crumbly material that other organisms can finish recycling. Although the process is slow compared with natural breakdown of organic matter, it demonstrates that evolution can adapt even to human-made challenges. With careful guidance, researchers hope to refine these enzymes into powerful tools for reducing waste and reclaiming valuable resources trapped inside stubborn synthetic debris. Certain microbes participate in the formation of soil structure. Healthy soil is not merely a mixture of sand, silt, clay, and organic matter. It is a dynamic fabric woven together by microbial action. Bacteria and fungi secrete sticky compounds that bind particles into stable aggregates, preventing erosion and helping soil hold water. Within these aggregates, pores form where air and moisture circulate freely, creating tiny chambers that shelter seeds, roots, and microscopic animals. Some microbes coat mineral grains with biofilms that act like glue, while others dissolve or precipitate minerals that reshape texture. Their byproducts, including polysaccharides and organic acids, influence how nutrients cling to or release from soil particles. When roots move through the ground, they depend on these microbial structures to maintain the right balance of firmness and softness. Without this hidden labor, soil would collapse into compacted layers or drift away as dust. The living architecture created by microbes confers remarkable resilience, allowing landscapes to absorb rain, support plants, and sustain entire ecosystems. Some microbes emit gases that shape the chemistry of the air. In wetlands, oceans, forests, and soils, countless microbes generate gases as natural products of their metabolism. Some release carbon-containing compounds into the atmosphere, while others produce nitrogen-based gases that influence cloud formation or rainfall. In certain low-oxygen environments, archaea generate methane that rises through sediments and disperses into the air, though other microbes intercept much of it before it escapes. In oxygen-rich settings, bacteria produce trace gases that participate in reactions creating or breaking down atmospheric ozone. These gaseous exchanges occur on scales so vast that they influence climate patterns and air quality across regions. Even in forests, where trees dominate the landscape, microbial respiration contributes significantly to carbon cycling, breathing out carbon dioxide at night or in shaded soil. When considered together, these emissions form a complex atmospheric dialogue. Tiny organisms, each completing ordinary metabolic tasks, collectively help regulate the composition of the sky and shape environmental conditions for every larger creature. A few microbes create bioelectric fields around their cells. Certain bacteria generate electrical gradients that extend beyond their membranes, creating faint but measurable fields in the surrounding environment. They achieve this by moving charged ions in tightly regulated patterns, or by releasing redox-active molecules that shuttle electrons into nearby water or sediment. These fields influence how neighboring cells behave. Some microbes swim toward or away from electrical signals, while others adjust their metabolism based on the local charge landscape. In dense biofilms, overlapping fields form intricate networks where electrical pulses travel through the community like messages. These pulses coordinate activity when resources suddenly change or stress appears. The phenomenon blurs the boundary between chemical and electrical communication, showing that microbes do not rely solely on molecular signals. They also use subtle electrical cues to organize growth, guide movement, and synchronize responses. These living electrical systems demonstrate that nature has engineered conductive communication long before humans built wires or circuits. Certain microbes can withstand repeated sterilization attempts. In hospital settings, industrial facilities, and research laboratories, cleaning routines aim to eliminate all microbial life. Yet some species survive even intense heat, strong oxidizers, or high radiation. They accomplish this by entering dormant states where metabolic activity nearly ceases. Their protective coats thicken, membranes dehydrate, and enzymes fold into stable configurations that resist chemical damage. When exposed to ultraviolet light or chemical disinfectants, their DNA is damaged but not destroyed. Once conditions improve, Efficient repair systems replace broken nucleotides and restore functional genes. These microbes can endure cycles of destruction and recovery that would annihilate most organisms. Their stubborn survival poses challenges for sanitation efforts, but it also sheds light on fundamental questions about life's limits. They help scientists study how biological molecules can endure extreme stress, revealing strategies for long-term preservation that may prove useful in medicine, food technology, and the search for life in harsh, extraterrestrial environments. Some microbes turn sunlight into heat with unusual efficiency. Unlike plants that channel solar energy into chemical bonds, certain microbes absorb light and convert it almost immediately into gentle warmth. They achieve this using pigments that capture photons and release the energy as molecular vibrations rather than storing it. These microbes often live in cold water or shaded environments where generating warmth offers a competitive advantage. The heat they produce can keep cellular machinery flexible, allowing enzymes to operate efficiently even at low temperatures. In microbial mats, these heat-generating organisms warm their immediate surroundings, creating microclimates that support neighboring species. The pigments responsible for this conversion often appear reddish or brown and may have evolved originally as protective shields against harmful light. Over time, microbes adapted them into specialized heaters that maintain a stable internal environment. Their strategy illustrates that sunlight can be used not only for building energy-rich molecules, but also for simple thermal regulation on a microscopic scale. A few microbes feed on simple molecules found in ancient rocks. Deep within Earth's crust, cracks and pores hold water that has been trapped for immense stretches of time. In these secluded places, microbes survive on hydrogen, methane, or sulfate released by slow reactions between water and ancient minerals. They may live without any contact with the surface, drawing all necessary energy from these geological processes. Their metabolism unfolds at a pace that reflects the scarcity of resources. Some cells might divide only a handful of times over spans that feel more geological than biological. These communities reveal that life can persist in near isolation, dependent entirely on the chemistry of rocks. Their presence suggests that planets with thick crusts and subsurface water could host similar deep biospheres. The microbe's ability to use such simple molecules demonstrates that complex ecosystems can spring from minimal ingredients, given enough time and stable geological activity. Certain microbes maintain symbiotic relationships with coral. Inside coral tissues, photosynthetic microbes provide essential nutrients that fuel reef growth. These tiny partners absorb sunlight and convert carbon dioxide into sugars, which they share with their coral hosts. In return, corals supply them with nitrogen, phosphorus, and a protected habitat near sunlit waters. This relationship supports the vivid colors and towering structures of coral reefs. When environmental stress such as warming disrupts the partnership, corals expel their microbial partners, leading to bleaching and potential collapse of the reef. If under the right conditions, the symbiosis can be re-established. Some microbial strains even enhance coral resilience by producing antioxidants or stress-relieving compounds. The intricate exchange of nutrients, protection, and chemical signals highlights the interdependence of these organisms. Coral reefs, often thought of as animal structures, are fundamentally shaped by the microscopic friends living within their tissues. Some microbes influence Earth's climate through subtle chemical pathways. Across oceans, soils, and forests, microbes manage countless reactions that release or consume gases affecting global temperatures. In marine environments, certain planktonic microbes produce compounds that rise into the atmosphere and help form cloud droplets. More clouds can reflect sunlight and cool the planet. In soils, Microbial decomposers regulate the rate at which carbon dioxide enters the air by controlling how quickly organic matter decays. In wetlands, archaea produce methane, while neighboring microbes intercept and consume part of it, creating a delicate balance that influences the strength of this greenhouse gas. Even in deserts, microbes living on rock surfaces modulate how much carbon those surfaces absorb. These tiny chemical shifts accumulate into planetary-scale consequences. Their influence underscores a central truth. The climate system is not shaped solely by oceans, winds, and forests. Countless microscopic chemists help determine the temperature of the world. A few microbes can extract valuable elements from ore. In mining environments, tailings piles, and naturally mineral-rich soils, certain microbes cling to ore particles and release acids or oxidizing compounds that dissolve tightly bound metals. They draw energy from these reactions, using the released electrons for growth. As the ore dissolves, metals such as copper, nickel, or gold become mobilized and can be recovered from solution. This biological extraction avoids the need for intense heat or harsh chemicals. In some places, microbial processing is responsible for a large share of metal release from natural deposits. These microbes often thrive in acidic conditions that would destroy most cells, yet they use these extreme environments to access energy sources unavailable elsewhere. Their capacity to unlock valuable elements demonstrates the surprising reach of microbial metabolism. They turn inert rocks into sources of nourishment and, in the process, carry out mining on a scale far more subtle and patient than human industry. Certain microbes travel on dust across entire continents. When winds lift fine dust from deserts, dried lake beds, farm fields, and forest floors, countless dormant microbes rise with it. These tiny passengers cling to clay particles, grains of silt, and fragments of organic matter that drift upward into powerful air currents. High in the atmosphere, where temperatures drop and ultraviolet light intensifies, most life forms would perish quickly. Yet many of these microbes endure by entering dehydrated resting states that shield their DNA and stabilize their proteins. Some wrap themselves in thick protective coats, while others accumulate sugars that preserve internal structures like glass. Tarried by winds that can cross oceans and mountain ranges, they eventually fall with rain, snowfall, or settling dust. When they land, the first touch of moisture awakens them, allowing them to resume metabolism after journeys that may span thousands of kilometers. Their global dispersal alters microbial communities far from their origins, influencing soil fertility, plant health, and even the chemistry of high-altitude cloud layers. These travelers demonstrate that microscopic life is not confined by borders or landscapes. It rides the invisible highways of the sky. Some microbes thrive inside plant roots and boost their survival. Beneath the soil surface, a complex alliance unfolds between plant roots and specialized microbes. These microbial partners gather near root tips where plants release sugars and organic acids that attract beneficial species. Once settled, the microbes produce enzymes that unlock nutrients from soil particles, converting them into forms that plants can absorb. Some solubilize phosphorus, while others capture trace metals or release small molecules that encourage root branching. Certain species even help plants endure drought by stimulating thicker root walls or by producing compounds that reduce water loss. In return, the plant supplies constant nourishment, turning its underground tissues into a warm, nutrient-rich refuge. During times of stress, these microbes soften the impact of salinity, toxins, or temperature extremes. Their presence often determines whether seedlings flourish or fail. The partnership forms an underground support system where chemical messages and shared benefits pass continuously between root and microbe, sustaining ecosystems from the soil upward. A few microbes form protective shields made of minerals. In aquatic environments, hot springs, and deep sediments, some microbes draw dissolved ions from the surrounding water and precipitate them into crystalline shells. These mineral coatings can be thin and delicate or thick and armor-like, depending on the species. The formation process begins at the cell surface, where organic molecules act as templates that guide mineral growth into repeating patterns. As more ions accumulate, the coating thickens, forming a rigid barrier that protects the cell from predators, toxins, or sharp environmental shifts. Some shells reflect harmful light, while others resist high acidity or heavy metal exposure. When the microbe divides, the offspring often inherit fragments of the shell, which quickly grow into full coverings. Over time, abandoned shells accumulate in sediments as tiny fossils that reveal the microbial architects that once lived there. These mineral shields demonstrate how living cells can repurpose geological materials, crafting their own micro-scale armor with precision and elegance. Certain microbes can digest biofilms created by rivals. Biofilms are tough, slimy structures that protect microbial communities from drying, toxins, and grazing predators. Yet not all microbes accept their neighbors' fortresses as impenetrable. Some species have evolved enzymes that break apart the sticky polymers holding rival biofilms together. They dissolve the gel like matrix, exposing the underlying cells to attack or flushing them into the surrounding environment, where they become easy targets. These enzymes may slice through long carbohydrate chains, degrade structural proteins, or disrupt chemical cross-linking. Once the biofilm weakens, the invading microbes move into the cleared space, colonizing regions that were previously defended. In natural ecosystems, this constant competition shapes the distribution of species across rocks, plant surfaces, and sediments. It also influences the ebb and flow of microbial succession, determining which groups dominate at different times. The ability to digest another organism's shelter reveals a form of microscopic siege warfare, fought through chemistry rather than weapons. Some microbes produce natural antivirals that protect their communities. In dense microbial neighborhoods, viruses pose a constant threat. To defend themselves, some bacteria and fungi secrete molecules that block viral attachment or interfere with viral replication. These natural antivirals can bind to viral coats, preventing them from docking onto cell surfaces, or they can disrupt early steps of infection before viral genetic material takes hold. In some biofilms, antiviral compounds spread through thin channels, forming protective halos around vulnerable cells. When viruses fail to penetrate these zones, the community prospers while neighboring unprotected groups suffer. These antiviral molecules often arise from ancient evolutionary arms races, where survival depended on disabling common viral predators. Modern research has revealed that some of these microbial antivirals resemble compounds used in medicine, although microbes have been crafting and refining them for millions of years. Their quiet defenses highlight the complexity of interactions that unfold at microscopic scales, where chemistry becomes both shield and sword. A few microbes thrive inside the bodies of deep-sea creatures. In the cold darkness of ocean trenches and hydrothermal vent regions, giant tube worms, clams, and other deep-sea animals rely on microbial tenants living inside their tissues. These microbes convert chemicals such as hydrogen sulfide or methane into organic molecules that feed their hosts. In return, the animal circulates these dissolved chemicals through special organs where the microbes reside. The partnership replaces the need for traditional feeding, allowing entire communities of deep-sea animals to flourish without sunlight or plant-based food webs. The microbes maintain remarkable stability despite fluctuating temperatures, pressures, and chemical concentrations. They adjust their metabolism to match the host's needs, increasing production during growth phases and slowing during lean periods. This mutualism represents a profound adaptation to extreme environments. Without these microbial chemists, deep-sea ecosystems built around vent fluids and methane seeps would collapse entirely. Certain microbes modify their surroundings with chemical sculpting. In soils, caves, and aquatic sediments, some microbes reshape their environments by releasing acids, alkalis, or metal-binding compounds. These chemicals dissolve certain minerals while encouraging others to crystallize. Over time, the microbes carve channels, pockets, and pits into rock surfaces, transforming smooth walls into textured landscapes. In caves, microbial acids contribute to the formation of intricate etchings and unusual mineral patterns. In soils, chemical sculpting alters particle sizes and porosity, influencing how water flows and where nutrients accumulate. Some microbes create microchambers within sediments that function as personalized habitats, complete with controlled airflow and nutrient gradients. These modifications persist long after the microbes move on, leaving evidence of their activity in the form of subtly reshaped geology. Their chemical artistry demonstrates that life does not merely adapt to environments. It also alters them with steady, deliberate strokes. Some microbes can adapt to new environments within hours. While many organisms require generations to adjust to new conditions, certain bacteria alter their physiology almost immediately. When exposed to fresh nutrients, unfamiliar toxins, or changes in temperature, they activate rapid response systems that shift gene expression patterns. Cells may boost production of protective proteins, remodel membrane lipids, or switch to entirely different metabolic pathways. Some microbes carry genetic circuits capable of sensing chemical signatures and flipping entire networks of genes on or off with stunning speed. Others engage in horizontal gene transfer, acquiring new capabilities from neighboring species in real time. These rapid transformations allow them to colonize new habitats long before competitors can adjust. This agility explains how microbes spread through oceans, soils, and animal bodies with such efficiency. Their ability to reprogram themselves on the fly makes them some of the most versatile organisms on the planet. A few microbes are so small that entire colonies could fit inside a single cell of a larger organism. Among the tiniest known bacteria are cells barely larger than the ribosomes found in other species. These microbes contain only the most essential molecular machinery, packed into volumes so small that their internal components crowd together in tightly organized arrangements. Despite their minute size, they divide, metabolize, and respond to environmental signals with precision. Some live attached to the surfaces of larger microbes, relying on their hosts to supply compounds they cannot synthesize. Others drift freely in nutrient-rich waters, forming colonies that resemble faint clouds under a microscope. A single cell of a larger organism could easily hold hundreds or thousands of these microbes, yet each remains a fully autonomous life form. Their existence pushes the lower boundary of biological complexity, revealing how little material is required for life's core processes. Certain microbes are capable of transforming entire ecosystems over time. Across wetlands, coral reefs, forests, and ocean basins, microbial communities shape the environments around them in sweeping and long-lasting ways. They influence nutrient cycles by determining whether carbon is locked in sediments or released into the atmosphere. They regulate soil fertility by controlling the availability of nitrogen and phosphorus. In aquatic systems, blooms of photosynthetic microbes can increase oxygen levels, while other groups may trigger oxygen depletion by accelerating decay. Microbes also guide the formation of minerals, stabilize sediments, and support the growth of plants and animals through complex symbioses. Over centuries, these cumulative changes alter landscapes, reshape coastlines, and even drive evolutionary pathways for larger organisms. Every ecosystem carries the signature of its microbial architects. Their patient chemistry underlies the flourishing or fading of entire habitats, proving that the smallest life forms often hold the greatest influence. As our quiet journey through the microbial world comes to a close, let your thoughts drift gently toward the tiny lives that shape every corner of our planet. These unseen companions work in silence, weaving the chemistry of oceans and forests, guiding the rhythms of soil and sky, and reminding us that even the smallest creatures can hold astonishing power. Knowing they are out there, busy in their hidden realms, makes the world feel richer and more alive. If this peaceful wander through the microscopic universe brought you calm or sparked a little wonder, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, carried softly on the same calming current that brought you. But now, the night is waiting. Let your breath slow. Let your body grow still. And let the gentle hum of the unseen world guide you into deep and restful sleep. Good night.